Alright, so in this video, I'm just going to be going over the PCR in-class activity that you guys did in class. So yeah, let's just get into it. For number one, it's basically just asking what are the four main steps of PCR, and then like for in each step, which of the enzymes or reagents are more involved in, or like which ones are required. So yeah, to make it easy, I'm just going to go over enzymes and reagents first and kind of give you guys some kind of review also. So enzymes and reagents. Most important thing that we really need for PCR is DNA template, because just like DNA replication, we need a template to make copies, right? Because we cannot make a copy without a template. So yeah, DNA template is the most important thing. Next one, we need primers. But here's the thing. In DNA replication, we use RNA primers, but in PCR, we use DNA primers. There's a lot of multiple reasons like why we use DNA instead of RNA primers for PCR. One thing is because we don't really want to use a lot of enzyme too, or like because enzymes are not cheap. They're not cheap, so we can just make DNA primers that are like specifically complementary to the DNA strand instead. That way, like we're more efficient and we don't have to remove the RNA primer using another polymerase, stuff like that, So it's because it's budget friendly and more efficient. That's why we use DNA primer in PCR. Next one is Taq Polymerase. Taq Polymerase is just another type of DNA polymerase, but we use Taq Polymerase in PCR because it can withstand high temperatures. And during denaturing, the temperature is so high that some enzymes, they could be deactivated or like they could start deforming. So Taq polymerase is very heat resistant. That's why we use Taq polymerase for PCR. And then last but not the least, we need nucleotides, because what is nucleotides used for? It's used for elongating, right? Like even if we have Taq polymerase, if we don't have the nucleotides, then we cannot elongate the strand. So yeah, these are the four main things that we need for PCR. So I just want to drop this here because in an actual PCR lab, or like in an actual lab, all of this will go in just one test tube. Like you would put all of this in one test tube, and then when it's asking for this question, like for each step, indicate what enzymes or reagents are required, it's basically asking like which of these enzymes or reagents are most involved on each step. It's not asking like, oh, in Denature, what should we add? In annealing, what should we add? In synthesis, what should we add? Like all of this will just be added in one test tube at the very beginning. And then in each step, each of them are just involved more, or like the most involved in each one. So Denature, let's start with number one, Denature. If you remember, in DNA replication, we have to unzip the DNA, right? So for PCR, we do that by denaturing the DNA strands. And my own definition for it is basically unzipping DNA with a high temperature, because with high temperature, hydrogen bonds can be broken. So if we broke the hydrogen bonds between the bases, it will just unzip it like what helicase does, right? So when it comes to reagents, the most involved reagent in denaturing is the DNA template. That's basically it. That's the only thing we're dealing during denature. Second one is anneal. It happens at 55 degrees Celsius. And think of this as cooling down. Like even, I don't know if you have other ways of trying to define it, but for me, my way of definition for annealing is cooling down the system so that DNA primers can be added. Because DNA primers are not synthesized by the Taq polymerase. It is just it already has its own complementary sequence to the DNA. So the logic here is that if we cool it down enough, that means the primer can just, bond to the DNA template. And then once it's bonded to the DNA template, we start a synthesis. and it happens at 72 degrees Celsius. Why does it do that? It's because Taq Polymerase like works more efficient at that temperature. So yeah. So because we have the DNA primers, that means the Taq Polymerase knows where to start. And once it knows where to start, it can just keep adding the nucleotides. So yeah, these are basically like the reagents and enzymes required or like most involved in each step. And then last but not the least, number 4, repeat. Always include this, especially in your exam, always include repeat, because repeat in PCR we don't just make one copy of it. We normally make multiple copies of the DNA template so that we can study it. So repeat is basically just depending on how many number of cycles you put in there. So yeah, this is basically just number 1. I just made it longer because I wanted to explain what these are. Alright, so let's go to number 2. Number 2 is basically just, I will just shorten it. So number 2 is, what it's asking for is that we have a primer that is 24 nucleotides long. And then, where would it be in a DNA template? And then, where would the elongation start? And then what direction would it be? So that's basically it. So if you understood DNA replication, this will like basically be easy for you. Remember, we always start from 5' to 3', right? We always read 5' to 3'. Same way here, 5' to 3'. So when we're making strands or new strands or daughter strands, we always start with 5' to 3' also. And it's always anti-parallel to the DNA strand, right? So if we're gonna do this number 2, it says we have a 24 nucleotide long primer. So remember, this is 5' to 3', right? That means my complementary strand will be like around here. Let's say this is 24 nucleotides long. So it is anti-parallel. And then once PCR starts, remember, we can only add at the 3' end. So this is your primer, and it is basically just your elongation part or your synthesis part. So yeah, it's just as basic as this. Like, this is basically what it's asking for. Like, where would the primer be and what part would the elongation start, right? So primers would start always at 5' to 3', and then at the 3' end of the primer is where elongation will start. And then this is your new 3' end. Same way at the bottom. I know this is 3', this is 5', so I have to be anti-parallel to it, and we always start at 5' to 3'. So that means my primer will be around here. Let's say this is 24 nucleotides long. So that means this is where my primers will be, right? And then, since I know this is the 3' end, this is where my elongation will start. So yeah, this is basically just number 2. This is what it's asking for. And let's go to number 3. Number 3 is just basic math, or like, it's not basic, I don't want to say basic, but it's just math that you gotta do. Let me try to visualize in a way that you would, I guess it would help you understand better. So it says we start with 100 molecules of DNA, and then how many molecules will you have after? So let's visualize first. Let's say I start with just one molecule, right? When we say a molecule of DNA, we're talking about this whole thing, like this double-stranded DNA. We're not talking about just one strand. We're talking about the whole strand of the DNA. So yeah, let's say we have one molecule. On the first cycle, remember, both strands will be denatured, right? So we will have 2 separate strands. And then annealing and synthesis will happen. So that means we have 2 new daughter strands, right? And this is only during the first cycle. And then Let's say that's the second cycle, right? What's the next cycle? Next cycle is basically the same thing. You will denature this. So that means I will have something like this and then one at the bottom. Same way over here. You will denature it. Annealing and synthesis will happen. So that means I will have new strands again. New DNA molecules. And this happens during, let's say, during the second cycle. Now, if you get the pattern, this is basically what it's asking for. So if we start with one molecule, that means after 2 cycles, we'll have 4 molecules, right? 4 molecules of DNA. Now, if we apply that with question number 3, let's say we have 100 molecules. Think of it this way. We start with 100 molecules, right? 100 molecules. And then at the first cycle, we're going to have 100 molecules over here, 100 molecules, because think of it this way. This is 100, right? And then after the first cycle, we're going to have 100 also here. 100 molecules here, 100 molecules. So that's 200. Second cycle is going to be another 100 here, 100 here, 100 here, 100 here. So in the end, the amount of molecules that we have for 2 cycles is equal to 400 molecules of DNA. Yeah, so if we're gonna answer this, the first one is 400 molecules after 2 cycles. Now, the next one is 10 cycles, but we don't want to keep drawing this, right? We don't want to keep doing this, like this whole thing. We don't want to keep like, oh, let's draw 10 times 10 cycles of this. So the formula that we can use for this is basically the number of molecules multiplied by the number of strands produced, or like the number of basically the number of strands produced after denaturing, and we produce only 2 strands, and then to the power of the cycle, to the number of cycles. So this is the mathematical formula for solving this type of problem. So if we just apply it to this 10 cycles of PCR, basically if we start with 100, then we just multiply it by 2 to the power of 10. And then just put that in your calculator and that will be your answer. Same thing for letter C. It's just 100 times 2 to the power of 36. Because, you know, like the formula is just number of molecules times 2 to the power of the cycles. And then same logic can be applied for number 4. But in number 4, here's the prompt. We only have 1 primer. Right? So let me try to visualize it to you. So in this one, in number 3, we were able to make 2 strands because we have enough primers to make strands, right? So for this one, let's say 2 primers are working. That means we can make this new strand over here, and then these strands are over here. But for number 4, what it's asking us is that we only have 1 primer. So we will start with 1 molecule again. 1 molecule, we're gonna have 2 strands, right? Because once we denature it, we're basically separating it into 2 strands. But here's the thing. Only 1 primer is gonna work. So that means only 1 of the strands will have the primer. And remember, we need primers to elongate the new strand. So in this case, only this strand will be able to be elongated. Right? So that means we started with one molecule, we're going to end up with 1 molecule again. And then this one will just be lost. And then let's say we went to we had another cycle. Same thing, it's going to be denatured. right? But we only have one primer. So that means only one of these two will have a primer and will be able to be elongated. Let's say only the bottom one was elongated. So that means we still have the same amount of what we started, because this strand over here will not be elongated or will not be replicated because it doesn't only one primer is working. And then we cannot replicate this if there's no primer, right? So this one will be lost, and we end up with the same amount as we started. So with one molecule, we end up with just making one molecule again, and then one molecule again. And then you can just keep repeating it. You will see the same pattern. You will just keep getting the same amount as you started. No matter if you started with 100 molecules, you're going to end up with 100 molecules again, and then 100 molecules again on the next cycle, and then so on and so forth. But if you want to make it the mathematical way, same thing. Number of molecules multiplied by the number of strands made. But in this case, we can only able to deal with one strand. So that means it will be 1 to the power of cycles. And 1 to the power of anything is just 1. So no matter if I have 100 times 1 to the power of, let's say, 1 million or 1,000, it will just equal 100 again. So yeah, that is number 4, and that's everything that's in this video.